Lab 5, Fetal Pig Dissection Part 3, Circulatory, Respiratory, and Nervous Systems by Sasha Pankertoff and Brenna Puglisi.
The focus of Lab 5 is to study the mammalian respiratory system. In the medical field, it is essential to understand how the respiratory system works in order to diagnose and treat patients with respiratory disorders and diseases. To correctly diagnose patients, it is vital to understand how a healthy respiratory system functions, which is indicated by the individuals who fall within 2 standard deviations of the mean of the population.
The respiratory and circulatory systems work together to maintain homeostasis of the body. The major organs involved in the circulatory system are the heart, arteries, veins, and capillaries. This system is responsible for transporting substances such as nutrients, gases, wastes, and hormones throughout the body. The circulatory system works together with the respiratory system to oxygenate the blood and remove carbon dioxide via the capillaries and alveoli inside the lungs. This maximizes the surface area in the lungs, allowing for rapid and efficient gas exchange.
A buffer is a compound in solution that resists change in pH when a quantity of hydride or hydroxide is added to the system. The carbonic acid bicarbonate buffer system in the blood exists to maintain a pH between 7.0 and 7.8. The major component of the extracellular bicarbonate buffer system is carbonic acid, which is formed by anhydrase in the red blood cells. Carbonic acid dissociates into bicarbonate and hydrogen ions. Hemoglobin binds the hydrogen ions to prevent an intracellular rise in pH, while the bicarbonate diffuses into the plasma. A change in blood pH results in a change in pH of all of the cells of the body, affecting function and catalytic activity of enzymes. Thus, it is important that this buffer system maintain a balanced pH.
In our lab, one of the experiments involved using lime water. Lime water is concentrated calcium hydroxide in solution. It is used as an indicator for carbon dioxide. Alone in solution, it is a clear liquid. When carbon dioxide is introduced, it reacts with the calcium hydroxide to form a calcium carbonate precipitate, causing the solution to turn cloudy.
Phenolphthalein is another indicator of the level of carbon dioxide. Under acidic conditions, phenolphthalein changes from a pink color to clear. When carbon dioxide is bubbled through a solution of phenolphthalein in water, carbonic acid is produced and is responsible for the color change that is observed. Phenolphthalein is a weak acid that can lose hydrogen ions in solution. The phenolphthalein molecule is colorless, while the phenolphthalein ion is pink. When a base is added to phenolphthalein, such as ammonia, the acid is deprotonated, resulting in ionization and the visible characteristic pink color, which is an indication of the presence of carbon dioxide.
Vital capacity is a measurement of the maximum volume of air that can be exchanged in lungs in one breath. Vital capacity is the sum of 3 components: tidal volume, inspiratory reserve volume, and expiratory reserve volume. Tidal volume is the amount of air exchanged during a regular breath. Inspiratory reserve volume is the amount of air that can be forcibly inhaled after normal inspiration. Expiratory volume is the amount of air that can be forcibly exhaled after normal expiration. Vital capacity is dependent on age, sex, and height, as the lungs of every individual are unique. Vital capacity can be measured using a device called a spirometer.
Several objectives guided the observers through their examination of the respiratory system during the completion of Lab 5. To begin, the acid-base balance was investigated to gain an understanding of the importance of equilibrium and homeostasis of the body. Secondly, the observers strived to utilize a spirometer to examine human lung capacity.
Before the experiment was conducted, several predictions and hypotheses were generated. Firstly, with regards to the lime water experiment, it was predicted that exhaled air would turn the lime water solution cloudy more rapidly than the room air. This difference in rates would be indicative of the amount of carbon dioxide bubbled through the solution.
Next, with regards to the phenolphthalein experiment, it was hypothesized that if room air was bubbled through a solution of phenolphthalein, it would contain less carbon dioxide than exhaled air. Additionally, post-exercise exhaled air would contain even more carbon dioxide than either resting exhaled air or the room air, causing the phenolphthalein solution to turn from pink to clear at a faster rate. The difference in carbon dioxide would be indicated by the quantity of drops of ammonia required to turn the solution from clear back to pink.
Lastly, with regards to the vital capacity experiment, it was predicted that the vital capacity of the subjects would be within the normal range of the class data, within 2 standard deviations of the mean.
Concerning our materials and methods, Lab 5 was completed according to the Fetal Pig Dissection Part 3, Circulatory, Respiratory, and Nervous Systems procedure, provided by M. Buchholz and found in Worcester Polytechnic Institute. No procedural changes were made during the completion of this experiment.
The first experiment involved bubbling room and exhaled air, both containing carbon dioxide, through a solution of lime water. The results were consistent with the original predictions that were made before conducting the experiment, as exhaled air contained more carbon dioxide than room air. Table 1 shows that the room air took over a minute of bubbling to turn the solution cloudy, while the exhaled air took only 18 seconds to observe a change, becoming cloudiest at 35 seconds. These rates of change indicate that there is more carbon dioxide in the exhaled air relative to the room air. This greater concentration of carbon dioxide in exhaled air drives the equilibrium to the right, yielding the calcium carbonate precipitate at an increased rate compared to that of room air.
Our next experiment involved investigating the effects of exercise on carbon dioxide concentration. Different types of air were bubbled through a solution of phenolphthalein, turning the solution from pink to clear. Ammonia was added dropwise until the original pink hue returned to the solution, indicating the relative amount of carbon dioxide in the solution. The results of this experiment provide evidence to support the validity of our hypothesis. As seen in Table 2, prior to exercise, it took only 11 drops of ammonia to deprotonate the phenolphthalein, causing the solution to turn pink. Post-exercise, the solution required 13 drops to observe an equivalent color change. This difference in volume of base required to deprotonate the phenolphthalein indicates that there was originally a greater concentration of carbon dioxide in post-exercise air in solution that had protonated the phenolphthalein molecules. In comparison, there was less carbon dioxide in the pre-exercise air, thus requiring fewer drops of ammonia to turn the solution back to its original pink hue. Therefore, as predicted, post-exercise air contained more carbon dioxide than pre-exercise air.
By referring to the results of the single-factor ANOVA performed on the class and section data, it is possible to conclude if exercise has a statistically significant effect on carbon dioxide production. The null hypothesis for the experiment stated, variance in the physical activity of a subject will not affect the production of carbon dioxide. The alternative hypothesis stated, variance in the physical activity of a subject will affect the production of carbon dioxide. Figure 1 shows the single-factor ANOVA for the number of drops of ammonia required to induce a color change for the class data. The analysis resulted in a p-value of 3.86 times 10 to the negative 11. This value is less than 0.05. Thus, the null hypothesis is rejected and the alternative hypothesis is accepted. Variance in the physical activity of a subject will affect the production of carbon dioxide. This infinitesimally small p-value indicates that there is a statistically significant difference between groups and only an extremely small possibility that the variance observed is due to chance alone. This statistical analysis provides evidence to support the original hypothesis that post-exercise air indeed contains more carbon dioxide than resting exhaled air.
In the last experiment of Lab 5, vital capacity of the observer's lungs was measured using a spirometer. Observer 1, a female, had an average vital capacity of 3.63 liters, while the male observer had an average of 4.73 liters, as seen in Table 3. The standard vital capacity for a 20-year-old, 160 centimeter tall female is 3.40 liters, while the standard vital capacity for a 20-year-old, 170 centimeter male is 4.47 liters. Both of the observers' vital capacities were above the average for their height and sex. This indicates that they have efficiently functioning respiratory systems that allow for a large volume of exchangeable air in the lungs. Therefore, it was predicted that when the mean and standard deviation of the class vital capacity was generated, the average vital capacity of the observers would be within 2 standard deviations of the mean. Table 3 also documents the calculated mean and standard deviation for the class vital capacity data. The mean vital capacity for the class was 3.46 liters, and the standard deviation was 0.78. Being healthy individuals, both of the observers' vital capacities were in normal range as they were within 2 standard deviations of the mean.
Our general conclusions were that room air contains less carbon dioxide than exhaled air, and that post-exercise air contains a greater concentration of carbon dioxide than normally respired air.